Permeable membranes are one of the key components of hydrogen fuel cells, one of the main technologies being explored to provide clean energy for industrial transportation. In this video, we'll analyze a fuel cell membrane that has already been in real-world operation. Such extended use can result in the accumulation of contaminants on the membrane surface, which in turn compromises its performance and functionality. We will adhere to ISO and VDA standards to perform chemical identification using FTIR microscopy in order to trace the source of the contamination. First, a visual overview image of the fuel cell membrane is captured using the Opus software. The camera clearly reveals the membrane stripe topography. The multi-tile focus fusion function is used to generate a fully focused image along the Z-axis within the defined parameters. Next, the FPA infrared imaging detector is selected to generate a true FTIR image of the sample surface. Using ATR mode allows for high-resolution analysis at 1.25 micrometer spatial resolution. Then the area of interest is defined for detailed analysis. We decided for 6x12 FPA imaging tiles. The FTIR imaging begins after assigning a name and file path for the experiment. Each tile will capture 1024 spectra across an area of 1600 square micrometer. The measurement is finished and we receive an infrared image containing over 74,000 spectra. By clicking on the bright reddish spots, spectra rich in carbonyl groups are revealed, indicating areas of polymeric contamination on the membrane. Now, an exemplary spectrum from the contaminated area is used to identify its chemical composition. To facilitate identification, we use the machine learning-based composition aid, or AID function, which performs both single and multi-component analysis automatically. In the end, AID identifies the contaminant as resin. This information can be vital to optimize production properties or develop new, improved materials. That's it. Thank you for watching.